In the second video, we're going to talk about the first step of protein synthesis. Protein synthesis is taking the code that's stored in your DNA molecule and taking it from this DNA and ultimately creating a protein. Now, we talked about how proteins do lots of different kinds of things. So proteins can become enzymes, proteins can become parts of hormones, proteins can make up melanin in your skin to give your skin different colors. It has a lot of different functions. And the function of the proteins depend on the code that is spelled out in your DNA molecules. So the first step to get that code out of the DNA and into a final protein is the process of transcription. Transcription starts in the nucleus, and it's done by an enzyme called RNA polymerase. So the way this works is RNA polymerase is going to break the GC-AT pairs of your DNA. It's going to split this molecule along its center so that we can see the code in the middle of this DNA. The place where that code is exposed is where we're going to start building our mRNA molecule. So just like before, everywhere that there's a G, we're going to add a C. Everywhere that there's a, can you help me real quick, Courtney? Can you hold this? Hold the phone. Just on here, thank you. Everywhere that there's a C in the DNA template, we're going to add a G nucleotide to the mRNA strand. We have a G, so we need a C molecule. We have a T, so we need an A molecule. We have an A, and you want to put a T molecule there, but we got to remember that we're making mRNA, and thymine is not part of mRNA. It's replaced with the base uracil. So that's one major difference between DNA and RNA. RNA only uses the base U, whereas DNA uses the base thymine. The other differences are that mRNA is a single-stranded molecule, whereas DNA has two strands. And there's also a difference in the sugar that makes up the backbone. So if you remember from the previous video, the deoxyribose, that gray molecule in the DNA backbone, is not present in RNA. It becomes a ribose sugar. So that's why DNA, deoxyribonucleic acid, starts with a D, and RNA, ribonucleic acid, starts with an R. So this process is going to continue until the entire mRNA strand is coded. So we're going to make a copy of this entire DNA molecule. And you'll see that this DNA is able to come back together and it continues to be stored in the nucleus. So we save our original copy of those instructions, and we can make lots of different protein molecules from the same DNA strand. Okay, so I'm going to finish the transcription of the mRNA, and then I'll start the next video on the second step of protein synthesis, translation.